This is for Biology 223 Genetics. We have an article here from PNAS, Proceedings of the National Academy of the Academy of Sciences, United States, published in 2016. Eric Kandel is well established in this field. This relates to text chapters on the regulation of gene expression in eukaryotes, also to some extent to the genetics of cancer. And although we don't cover genetic analysis of development. Roles for small non-coding RNAs in silencing of retrotransposons in the mammalian brain. So as in some other papers we've seen, there's this idea that the mammalian brain during development undergoes changes, not just developmental changes through epigenetic, but also genomic mosaicism, in this case caused not by these long, actively transcribed, late replicating or genes, but by retrotransposons. So non-coding RNAs, they're not being more specific than that. That's fine. It doesn't matter to them specifically. They don't want to state microRNAs or silencing RNAs. These non-coding RNAs, these small RNAs, the mechanism of how they work is a bit complicated, and how we even describe them is more often by their biogenesis, and they get into what they care about here. So let's start. First sentence seems to be a general description of phenomena of interest. Pee-wee interacting RNAs, piRNAs, long thought to be restricted to the germline. Long in this case is decade or two. Have recently been discovered in neurons of aplysia. That's the sea snail. That's a model of upper conditioning of memory, with a role in the epigenetic regulation of gene expression underlying long-term memory. Okay, so they're telling us this phenomenon that piRNAs, pee-wee, which was thought primarily to, and it's described here, that it's a big subject. It's a mechanism of transcriptional repression of transposable elements in the germline, and two important model systems, Drosophila and Cinerevis elegans, and Drosophila, which I'm a little more familiar with. There are these clusters of loci which seem to have collected sequence information about transposable elements, kind of like antiviral software, and use that to generate transcripts that are then processed, and through this amplification, ping pong amplification, go and bind to transcripts from retrotransposons, and during transcription, which leads to recruitment of DNA silencing, transcriptional silencing via methylation and histone mechanism. So it's a quite interesting concept that in order for something to be silenced, it has to be actively transcribed, at least at low level. That's somewhat described here in the first paragraph. Pee-wee interacting RNAs are 26 to 32 nucleotide small non-coding RNAs that associate with the pee-wee clade of Argonaut family of proteins, and whose primary functions in mammals are associated with the silencing of transposons in the germline through de novo DNA methylation. Transposons constitute 44% of the human genome and could, when active, contribute to genetic heterogeneity, to alteration of behavior during adaptation and responses to stress. So the issue here is this genetic heterogeneity, this genetic mosaicism, and specifically interest here in the concept of the brain requires this genomic mosaicism in order to be functional, as well as when dysfunctional, when misregulated, creating mental illness. Somatic retrotransposition and it's associated insertional mutagenesis because, right, a retrotransposon, it's not just expressed, it can be reverse transcribed and insert somewhere else, and that kind of insertional mutagenesis have particular important implication for brain disorders and often associated with schizophrenia, Rett syndrome, I don't know what that is, and neurodegenerative disorders. And one might imagine cancer. Line 1, that's a long interspersed nuclear elements, oops, says so, family of retrotransposons in particular is active in the human and mouse hippocampus. Okay, so let's go here. We here ask whether PWE/piRNA, so they mean that they're considering them to be synonymous. If PWE is expressed, then piRNAs exist. If piRNAs are found, then PWE is expressed. We're also expressed and have functional roles in the mammalian brain. Large-scale RNA sequencing, this is a technological development of the last couple of decades, where sequencing directly, large-scale deep sequencing inexpensively, and also the computational technology to analyze it. Large-scale RNA sequencing and subsequent analysis of protein expression revealed the presence in brain of several piRNA biogenesis factors. So, we define piRNAs as resulting in the biogenesis, which include this Argonaute proteins. And so if we see the factors, then that's a signature that they probably exist, including a mouse PWE, here called milli, so it has its own name, as well as small RNAs, albeit at low levels, resembling conserved piRNAs in mouse testes, which is the germline where we expect them. Primarily line 1 retrotransposon derived. So this is a little bit complicated, whether, and later it says that in the paper that they don't see that this resembles the Drosophila system, in which are piRNA clusters to generate it. It's that the primary transcript, the direct transcript of the line is used to be turned into a piRNA and then going through the ping pong cycle to suppress the transcription at its own locus. So it's a somewhat different system. Despite the seemingly low expression, so even though they're very low expressed, of these putative piRNAs, single base pair CpG methylation analyses across the genome of milli-piRNA deficient mice, so that would be they've knocked out the gene, demonstrate that brain genomic DNA is preferentially hypomethylated, which is associated with activity at CpG. So we have these CpG islands in promoters and in gene suppressed. They're found in promoters, and when methylated, we expect transcriptional repression, and when undermethylated, hypomethylated, we expect association of euchromatin and transcriptional initiation. So we would expect normally that retrotransposons are methylated. So here they demonstrate the brain genomic DNA is preferentially hypomethylated within intergenic areas and line 1 promoter areas of the genome. It's a little bit oddly written. The way the sentence is written, at first I thought, well, do they mean that there are hypomethylation sites within the wild normal brain genomic DNA, or in the knockout mouse, the mutant that would be incapable of generating these piRNAs? And reading the article, that's also a little bit ambiguous. Looking at other things, it's clear that we have, and it's hinted at here, a little bit frustratingly, hinted that, yes, there is and this is the most interesting part of the paper in this area of research, that there is a role for retrotransposons, retrotransposition in normal brain development. And so any alteration of that, in this case of reducing the ability of piRNAs to repress retrotransposon, causes problems with brain function. But the way it's written here, you might think that hypomethylation sites are only in the mutant. No, I believe it's in the wild type. Furthermore, milli-mutant mice exhibit behavioral deficits such as hyperactivity and reduced anxiety. So that's to show that if this function, which is presumably leads to the absence of piRNAs, then there's a phenotypic, obvious phenotype of reduced anxiety, which is a bad thing for mice, being prey. Hyperactivity, which is also, they don't talk about it and you'd have to see other articles talk about the role in normal brains. It's hinted at here later. These results suggest that putative piRNAs exist in mammalian brain. That's the big finding that we have, and that's hinted at in the first sentence there, that Pee-wee interacting RNAs was thought to be a germline, primarily germline, or found in the germ. Here, they're found in the mammalian brain. And similar to the role of piRNAs in testes, they may be involved in silencing, presumably are involved in the silencing of retrotransposons. In the germline, it's particularly important because there's global methylation patterns and erasure, almost complete erasure and deposition of epigenetic marks in the process from gametogenesis through the zygote formation and early embryo that are very, very important in development here, which in brain have critical roles in contributing to genomic heterogeneity underlying, and these point out, these are not disease states, these are normal states, adaptation, stress response, and brain pathology. So the genomic heterogeneity that we see in the mammalian brain is associated with function and pathology. We also, function and dysfunction. We also describe the presence of another class of small RNAs, that's an important discovery, with features of endogenous small interfering RNAs, which may have taken over the role of invertebrate piRNAs, such as Drosophila, in their capacity to target both transposons as well as protein coding genes. Thus, RNA interference through gene and retrotransposon silencing previously encountered in the Plizia may also have potential roles in the human brain. So that's pretty wild stuff. Let's go through the significant statement here. It's a little bit easier to digest. Pee-wee interacting RNAs, piRNAs, a class of small non-coding RNAs whose primary function in mammals is silencing germline resident retrotransposons, especially in early embryogenesis when some of the other mechanisms are not available, are also expressed in the Eplizia brain, which is what started this interest, where they play roles in memory storage. So that's interesting. Memory storage, memory in the C cell, it's a model. It's a very well-established model. It's hard to imagine they have the same sort of memories as we do, but nonetheless, that's the experimental model that we have that we can work with and make strong findings. Transposons constitute a bulk of the genome, and when activated can cause genomic instability and variability by integrating into various gene loci, including those in the mammalian brain, where they can modulate the stress response and adaptive behavior and contribute to brain pathology. Also to normal function. They're hesitant to state that. And how controversial that idea is, it's much easier to talk about pathology than normal function here. We show that small RNAs with conserved piRNA-like features are present in the mouse brain. In addition, we describe another class of small non-coding RNAs with features of endogenous small interfering RNAs, which along with putative piRNAs could play critical roles by silencing brain-expressed retrotransposons, contributing to mammalian behavior, that is function. So these have important functional roles, and they're kind of skirting around things here in a provocative way, and this is a little bit frustrating for somebody who's outside this specific field, so they're unwilling to make these clear statements because a lot of it is controversial and speculative. And you might notice here some of the funding involves this part of the world, as well as very well-funded institutions in the industrialized liberal democracies. I will stop here. Thank you.